Assalamualaikum everybody, welcome to the fourth live parasito session. Today we'll be talking about Cryptosporidium hominis in detail. In my recent sessions on parasitology, I've talked about parasitology in general, then I talked about Entamoeba histolytica and Giardia lamblia. So today we'll be talking about Cryptosporidium, as I've mentioned earlier. It is nowadays called Cryptosporidium hominis, but it was formerly called as Cryptosporidium parvum. P-A-R-V-U-M, that was its spelling. So moving on to the first slide. Cryptosporidium is responsible for causing cryptosporidiosis. That's a diarrheal disease. This disease ends with oosis, and as I've mentioned in the first session on parasitology, that parasites are responsible for causing diseases which ends with aces or oses. In this case, it ends with oosis, so we can identify it as a parasitic disease. In case of Giardia lamblia, it was responsible for causing so it ended with aces, which meant disease. And Cryptosporidium hominis causes diarrhea that is most severe in immunocompromised patients, especially those with AIDS, acquired immunodeficiency syndrome. Is this slide clear to you all? We are going to talk about characteristics in a little, okay, so a little later. So if you've got any questions about the introduction of Cryptosporidium hominis, you guys can ask me. Till that time, I'll talk about its characteristics. Parasites are divided into two categories. One is protozoa that are single-celled organisms, and other ones are the metazoa, which are multi-celled organisms. So Cryptosporidium hominis being a protozoa, an intestinal protozoa, which may be responsible for causing infections in the intestine. Let me introduce you to easy for you guys to understand Cryptosporidium hominis in detail. Protozoa are unicellular organisms, which means they're single-celled. A uni means single, right? And they've got no cell wall around them. Normally, animal cells and plants, animal cells have a cell wall around them, not animal, but plant cells have a cell wall around them. Humans being kind of animals do not have cell wall around their cells. Also, the cells that do not have cell wall around them. For example, Giardia lamblia. As you can see down there, it is a pear-shaped parasite and has got no cell wall around it. And these protozoa form cysts in condition where they feel that these are not favorable to growth. And cysts are they're a stage in their life cycle which makes them resistant to such unfavorable environmental conditions. Unfavorable environmental conditions include desiccation, hot temperature, or very cold temperature, and other conditions like that. Now let's move on to the classification of proteins. Two things: movement and regions. Regions mean site of action. So based on movement, protozoa are further classified into four categories. The first one is Sarcodina. These are amoebas, and I've put a cute little amoeba there, if you can see. These move with the help of pseudopods. They do not have a flagella or a cilia, but they do have pseudopods, and because of these pseudopods, they've got irregular shape. The second one is Sporozoa. These are the protozoa that do not move, and they've got no pseudopods, no flagella, or no cilia. The third ones are Mastigophora, which includes flagellates, for example, Giardia lamblia. As their name shows that they will be moving with the help of what? Flagella. And the fourth one is ciliata. They've got cilia that help them to move. So these are termed as ciliates. Is that slide clear to you all? Or if you've got any questions, you can freely ask me in the comments. I've got a comment from Dr. Feroz Khan. Assalamualaikum, ma'am. Wa alaikum assalam. If you've got any question, you can ask me in the comments. Then we'll talk about the classification of parasites based on regions. Regions mean the site of action where the parasite will cause the infection. It has got 4 types when it comes to its classification based on regions. The first one, when a parasite infects the intestine, it will be intestinal parasite. In urogenital tract, it will be urogenital parasite. If it causes infection in tissue, it will be called as tissue parasite. And if it causes infection in blood, it will be termed as blood parasite. Are these slides clear to you all? Now we'll talk about the life cycle of Cryptosporidium hominis. Okay, we got a comment. The slides are not clear. There might be some internet connectivity problem. Alright guys, is the visual and the slide now clear? And what about audio? Is it okay now? You can tell me in the comments. I think audio might be clear, but there's some problem in the slides. Is this clear now? Okay, we'll start talking about Cryptosporidium hominis life cycle. So we've got a comment. Dr. Feroz Khan is saying it's better now. That's great, thank you. So let's start talking about life cycle. Okay, as we've talked about the stages in the life cycle of parasites, they've got 2 stages. One is trophozoite and the other one is cyst. But in the case of Cryptosporidium, it's kind of different. It has got different stages, like first is going to be oocyst. In some places it's pronounced as oocyst, but I like to pronounce it oocyst. You can go with the one you like. So oocysts are released, and when somebody eats or consumes a food or water that is contaminated with the feces, the human feces that was infected with cryptosporidiosis, the person will get the oocyst inside its body. And in the intestine, the oocyst will release sporozoites. On the right side, you can see this purple image. This is the oocyst of Cryptosporidium hominis, and it has got certain structures in it, and these are schizones and merozoites. We'll talk about them in just a moment. We've got a comment from Study Hour is saying, sound is better now, but a little noise. Okay, let me figure out what's causing the problem. Is that okay now? Okay, you guys can tell me if there's any problem in the visual or the sound. Let me check, is there something wrong with my mic? Is it okay now? Can you guys clearly hear me? I think the slides are not clear. Might be audio. Okay. Let's talk about the remaining life cycle of Cryptosporidium hominis. I'm sorry for the disturbance caused due to a visual and audio problem today. I'll try not to do so in upcoming sessions. Okay, we're talking about that oocysts release the sporozoites in the intestine. Why in the intestine? Because Cryptosporidium has got a site of action, and that's the intestine. We've got a comment from the big dog K. I'm sorry if I'm not pronouncing your name correctly. Yes, it is okay. Thank you. That's great if you guys can hear me clearly. So oocyst will release sporozoites. What are sporozoites? These are living cells that can infect host, which form trophozoites. And all of this is happening in the intestine. Several stages, the schizoids, merozoites, and the trophozoites are formed in the intestine, and eventually microgametes and macrogametes form. These unite to produce a zygote, which differentiates into oocyst. And oocyst is then passed in the feces, which is responsible for causing infection. Is this slide clear to you all, or if you've got any question, you can ask me. If it is related to Cryptosporidium, its introduction, protozoa, classification of parasitology, or whatever. Okay, we'll move on to the next slide. Meanwhile, if you have any question, you can ask me. I've got a comment from Study Hour. There may be an issue with your mic. Ma'am, you can take time to fix it if you want. Otherwise, it's okay. I can hear you. Okay, I'll fix it, but I think if it's better, that's great. Let's talk about the transmission of Cryptosporidium hominis. It has got a fecal-oral route of transmission, which means that the cryptosporidiosis will be transmitted to humans by consuming food or water that is contaminated with the oocysts of hominis habitat. It has got two hosts, humans, which are the primary hosts, and animals, which are the occasional hosts. For example, now let's life cycle. As I've said, that Cryptosporidium is acquired by fecal. I'm sorry, I slipped my tongue. The organism, the Cryptosporidium, is acquired by fecal oral cysts from either human sources or from animal sources, for example, cattle. These oocysts then exist in the small intestine, where the trophozoites are released, and also the other forms, the schizoids and the merozoites, right? And they attach to the gut wall, where and there is no invasion caused by the Cryptosporidium hominis. As I've said, it is responsible for causing diarrhea. Cryptosporidiosis is a diarrheal disease, but there is no toxin associated with the diarrhea. Is this slide clear to you all, or if you've got any questions, you can ask me. Moving on to the next slide, that is clinical findings. I'm sorry if the mic is still causing problem or the visual is not clear. I don't know why it's not clear. In last 3 sessions, everything was okay. I'll figure it out. Okay, let's talk about the clinical findings of the Cryptosporidium. As it causes diarrhea and the diarrhea is watery, that's not bloody, so it will be causing fluid loss, and because of fluid loss there will be weight loss. And diarrhea is related to the intestine, so it will cause abdominal cramps there and will cause vomiting. And as this is a diseased condition, so the patient will have slight fever. So we've got a comment from Dr. Feroz Khan, that is which type of diarrhea it causes. Okay, that is watery diarrhea, not bloody. I hope that answered your question. Now let's talk about the lab diagnosis. We'll have a Q&A session at the end of this lecture. If you've got any question, you can ask at that time too. We'll take the sample of feces for diagnosis the infection caused by Cryptosporidium. And we'll then go for microscopy and we'll perform acid-fast test prior to doing the microscopy. Okay, here I'm going to ask you guys a question. What is acid-fast stain? I'll give you just 5 seconds so you can quickly write it in the comments. What is acid-fast stain? It's quite easy, I know. Come on, write it. It's different than a gram staining, which is done for the bacteria, acid-fast stain is also like mycobacteria, but it is also done for Cryptosporidium hominis. I've got no comments regarding the acid-fast stain, so I guess I should tell you guys, acid-fast stain is a differential stain, and that is used to identify acid-fast bacteria. Acid-fast bacteria, those bacteria that only stain with a more acid-fast stain, and they do not stain with gram staining. Because those bacteria that are acid-fast stain, they've got certain characteristics, like they've got wax-like, nearly impermeable cell walls. But in this case, this is Cryptosporidium hominis, and it's a parasite, and it has got no cell wall, but still, it is an acid-fast parasite. Okay, now let me tell you a little about its morphology. As all the parasites have got cysts, and cysts are circular or oval in shape. Same goes for Cryptosporidium, but it has got certain different shapes for sporozoites, merozoites, and trophozoites. Is lab diagnosis clear to you guys? Or if you have any question, you can ask me. We've got a comment from Dr. Feroz is saying, in 5 seconds, it's difficult to answer. I'll take care. I'll give you 10 seconds next time so you guys can answer. We're going to have a Q&A session as this is the end of the session. So we've got another comment from Dr. Feroz that at least a minute. Okay, I'll take care. I'll give you a minute for a question. Okay, you guys can ask me your questions, and I'll also ask you a few questions about today's session so I get to know how much you've gained from today's session. Okay, ask me questions, or I should ask you a few questions. We've got a comment from Adi. Good evening, ma'am. Oh, Adi, you were present in Entamoeba histolytica session. I remember you. Let me ask you guys a question. But first, study hour is saying a mycology session, please. Definitely, I'll do a mycology session after the completion of mycology series on channel. I've recently started it, and I've uploaded its introduction video. If you haven't watched that, be sure to check it out. Okay, the question I was asking you was about what kind of diarrhea. Is it bloody? Is it non-bloody? That is caused by Cryptosporidium. So I'll wait for some time, not 5 for 10 seconds, so you can answer me. We've got a comment. Is this protozoa resistant to any water purification? That comment is by Dr. Feroz Khan. Yep, it is resistant to water purification techniques like chlorinated water. We can go for that. And certain other water purification methods like filtration that can remove the cyst. I hope that answered your question. Okay, now you guys are supposed to answer my question. Tell me what kind of diarrhea Cryptosporidium hominis causes. Is it watery? Is it bloody? Okay, I'm giving you some time so you can process that in your mind and answer me. Till that time, I'll ask you another question. Okay. Come on, guys, that's quite easy. Answer the question. I've mentioned that in the clinical findings section. If you have been, so you might know it, or if you joined it now, so you might get answer from somebody else. We've got a comment from study hour. 1 minute, please. Of course, I'm waiting for your answer. We've got another comment from John Kangwa Katongo. I'm sorry if I'm not pronouncing your name correctly. That's watery diarrhea, correct? That's good. We've got an answer from Dr. Feroz Khan. That is watery. Yep, that's correct. We've got an answer from study hour. That's watery diarrhea. That's great. Okay, another question I'm asking you is about the route of transmission. Tell me, what's the route of transmission for Cryptosporidium hominis? That's really easy. Or if you have got any questions about today's session, so you can ask me. I'm waiting for your answer, guys. Come on, answer me. The route of transmission for Cryptosporidium hominis. Okay, I'm waiting. That was quite easy. What's the route of transmission for Cryptosporidium hominis? We've got a comment from Dr. Feroz Khan that is fecal-oral. Yeah, that's great. You're correct. We've got another comment from Tech Capital Investment. He said, hi, hello, how are you doing? Adi is saying, ma'am, it's getting too much hair fall. I'm sorry, I didn't get you. What are you asking about your hair fall, or is this a question related to something else? I would recommend please stick to the questions related to today's session, so that would be good. We've got another comment from Dr. Feroz Khan. It's easy, but I have internet issue. No problem. Take your time. We've got another comment from Study Hour. Fecal oral maybe. Yep, you're right. Not maybe. It is actually. Adi is saying my hair fall, ma'am. Okay, that might be due to dandruff, might be due to the kind of shampoo you're using, might be due to weather conditions, might be due to the water. I've got another comment from Dr. Feroz Khan. Our ma'am is very hurry for another class. Are you talking about me? Nope, I'm not in a hurry. Take your time and answer the questions. Okay, I've asked you 2 questions. Now I'll ask you the third question. That is, tell me about other symptoms of cryptosporidiosis. I know that's an easy question, but I trust you. You guys can answer that. Okay. I think there might be. We've got another comment from John that is fever. Yep, you're correct. That's fever. Okay. I'm waiting for your answers, guys. Study hour is saying fever. Okay, tell me about other symptoms related to watery diarrhea caused by Cryptosporidium. John is saying weight loss. Yeah, that's great. I think you've gained so much from today's lecture, John. Anybody else? We've got a comment from Dr. Feroz Khan, loss of appetite, bloating, gas, fatigue, etc. Yep, these are related to diarrhea. Adi is saying, ma'am, I wasn't in the class. No worries, you can watch that session after it's done. Okay, John is saying abdominal cramps. You're correct. Okay, let me ask you another question about Cryptosporidium. Study hour is saying it's bloating. Yeah, but not mainly bloating. We can say weight loss, diarrhea, fever, vomiting, stuff like that. Study hour is saying vomiting. Yep, that's vomiting. Okay, Adi, you asked me about my goal. My goal is to help you guys in getting good knowledge and help you guys to scope better in your class. And if you've got any suggestions for me to put what kind of videos on the channel, you can tell me in the comments. Emmanuel said hi. I'm sorry if I didn't pronounce your name correctly. Okay, I'll ask you guys another question related to today's session, and then I think we can end the session. Okay, tell me about acid-fast stain. Okay, I asked you that question in the lab diagnosis section, and nobody answered at that time. But now you guys are supposed to answer it, right? What is acid-fast stain? Why we use it for Cryptosporidium hominis? Okay, tell me, guys. There might be some problem. If you have any question related to acid-fast stain, I'll tell you. Or I can ask you another question if you don't know about that. Or maybe there is an internet connectivity problem. Okay, I'm waiting for your answer, guys. Okay, we've got a comment from Adi. Gram-positive testing, maybe. I'm sorry, Adi. Gram-positive testing is used for bacteria, not for parasites. And acid-fast stain is used for bacteria, but also for this parasite. Dr. Feruzhan is saying this stain used because parasite has mycolic acid in the cell wall. Well, Cryptosporidium hominis has no cell wall, but it stains with this acid-fast stain, the modified quinone acid-fast stain. Okay, we've got a comment from study hour. Acid-fast stain identifies tuberculosis. Yep, because tuberculosis is a mycobacterium, and mycobacteria are acid-fast bacteria. You're correct, but today's session is about parasites, so please stick to the parasites. Dr. Feruzhan is saying net is weak, no problem. You can rewatch the session after it's done. Study hour is saying acid-fast stain is for bacteria. Yeah, they are for bacteria. The acid-fast bacteria, those that belong to mycobacteria, those that belong to the mycobacterium family or genus. I'm sorry, I'm slipping my tongue again. I don't know why, but even legends make mistake. That's okay. Okay, that is going to be the end of session from my side. If you guys have got any questions, you can ask me in the comments, or I'll end the session very soon. I'll wait for some time so you guys can ask your questions, and then I'll end the session. I've asked you 4 questions and you've guys answered them correctly. That's very great. I'm very proud of you guys, of all of you. Okay, Adi, now we're going to talk about the goals. You're asking about my goal, what kind of goal you want me to share. The goal of this educational channel. Why don't you share your goal, Adi? That would be great to know what's your purpose in your life. Okay, Dr. Feruzhan is saying, ma'am. Yes, what's your question? You can freely ask me in the comments. If you guys want the PDFs of today's session, you can send me an email on my email address. I'll mention that in the comments. That is medzukhruf. Okay, here you go. And you guys can also connect with me on my Instagram. That's with the handle medzukhruf. I'll mention that for you in the comments, so you can easily search that on Instagram and connect with me. We've got another comment. Dr. Feruzhan is first time to join the live. That's very great. Welcome. We've got another comment from Adi. Your ultimate goal in your life. Why are you stuck to the goals, Adi? Why don't you ask questions related to the sessions? My goal is to help people become successful in their life, especially medical students. I want them not to face those problems that I had back in the medical school, right? I've got another comment from Dr. Feroz Khan. Please, can you introduce yourself if you really don't mind? You guys know me. I'm Zukhruf, the owner of Medzukhruf channel. Okay, we've got another comment from John. It is a differential test for this kind of parasite because it is able to stain with acid-fast test. That's correct. You've answered really well. That's a really good answer. Good, John. Okay, I think that's it for today's session. It's almost 10:37 in my country. So I'm about to end the session. And happy Ramadan, guys. It was day 2 in our country. What about you? Okay, I think this is it. So I'll say the ending remarks for the session today, and I'll see you in the next sessions. I really enjoyed this session with you guys, and I hope you did so. I'll see you soon in the next session. Till then, Assalamualaikum.